Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing TIFR Biology question paper conducted in the year 2014. If you are new to my channel, please do like, share and subscribe. Coming to the next year 2014. First question. During photosynthesis, the source of oxygen is. So the source of oxygen during photosynthesis is water. Answer is option 1. How many molecules of double-stranded DNA does a Drosophila cell nucleus contain? So the diploid chromosome number, that is 2n of Drosophila, is 8. The total number of double-stranded DNA in all eukaryotic cell will be 2n chromosome number of that organism, or the diploid chromosome number of that organism. So it will be 8. An allele has different effects depending on whether it was inherited from the father or the mother. This is most likely due to genomic imprinting. Then next one. A cell membrane isolated from a eukaryote was analyzed for specific binding of hormone receptor by incubating a constant amount of membrane with varying concentration of H3 labeled hormone. Which of the following graphs best depicts the results? Now cell membrane, it was isolated from eukaryote and it was analyzed for specific binding of hormone receptor by hormone. Now what they did was they radio labeled the hormone. So radio labeling, it will tell us how many molecules of hormone got bound to the receptor. If there are 2 receptors, then 2 molecules of hormones would have bound. If there are 3 receptors, then 3 molecules of hormones would have bound. So the graph will be as shown in D, because there will be an increase in the beginning. That means the number of receptors will be free from the hormone bound, from the bound hormone. So when you add hormones to these particular receptors, they will catch the hormone and they will not catch. The hormones will bind to the receptor. So the graph will be as shown in D. There will be an increase in the beginning, and if all the receptors get bound by the hormone, then it will become saturated and with hormones, and it will become a steady state like this. So it will be a hyperbolic graph. So the answer here is option D. The next one. Polyoma virus, a DNA virus, causes tumor in nude mice. Nude mice do not have thymus because of the genetic defect, but not in normal mice. The best interpretation is that. So polyoma virus, it causes tumors only in nude mice and not in normal mice. And nude mice, it doesn't have thymus. It doesn't have thymus means T lymphocytes are not produced. So T cells, because T cells mature in the thymus and nude mice does not have T cells, so they cause cancer. So what do you infer from this? That T lymphocytes play an important role in the rejection of polyoma virus induced tumors. Answer is option C. Now which of the following sequence correctly describes cell cycle? So the cell cycle is G1, S, G2, then mitosis, cytokinesis, and again back to G1. So G1, G2, mitosis, cytokinesis. That is not the correct sequence. Mitosis, G1 phase, G2 phase, no. Cytokinesis, mitosis, G1 phase, G2, no. S phase, G2 phase, mitosis, cytokinesis, G1 phase. That is correct. So option D is the correct answer. Then next one. The genome of a virus has composition of 25% G, 25% C, 30% A and 20% U. The genome is, so it is U is present, so it is not going to be DNA. So it is RNA. Now you have to see whether it is double-stranded or single-stranded. Now G is equal to C. Okay, but A is not equal to U. So it cannot be double-stranded. It is single-stranded RNA. Answer is option C. Then next one. In the measles and stall DNA replication experiment, what percent of the DNA was composed of one light strand and one heavy strand after one generation of growth in N14 containing growth media? So first it was grown. First, as per the experiment, the DNA was grown in N15 medium. Then it was transferred to the N14 medium. Now in N14, the DNA strands get replicated, and you will have one strand which is N15 and one strand it is N14. Like that, you will get two copies of your DNA. So they have asked what percentage of the DNA was composed of one light strand and one heavy strand after one generation. After one generation, all the DNA strands will be one heavy and one light. So answer is option D, that is 100%. Then next one. If offspring of heterozygous parents A capital A small a at a single locus are 25% capital A, 50% capital A small a and 25% small a, then all of the following are true except the parents are diploid organism. Yes, the A allele is recessive lethal. If that was the case, then you would not have got 25% small a. The alleles assort independently. Yes, the gametes combine at random. That is also correct. So the answer here is option B. Then next one. In the resting state of a neuron, the axonal membrane is comparatively more permeable to K positive ions and nearly impermeable to Na positive ions. The resting state of a neuron is minus 70 MV. So the answer here is it is more permeable to K positive ions. Answer is option. Why do glycolipids exist almost exclusively on the exterior side but not on the cytoplasmic side of the cell membrane? That is because these glycolipids, glycosylation, it takes place in endoplasmic reticulum and Golgi. So carbohydrates are added only to lipids on the luminal side of the endoplasmic reticulum and Golgi. Answer is option B. Next one. You discover a single gene mutation which causes lack of pain sensations. This turns out to affect a sodium channel and thus block conduction in nerves that convey pain. So you develop a toxin to block the channel in the hope of making it a cure for pain. It doesn't block pain, though it binds and blocks the channel just fine even in the live organism. What might be a good reason why it failed to block pain? So there is a developmental requirement for the channel wiring up in the pain centers. Developmental requirement means from a very young age onwards. That is from the newborn onwards. There is developmental requirement for channel wiring up the pain centers. But if it is blocked after adulthood, pain centers are intact and alternative pathways can stimulate them. So the answer here is option A. In adulthood, wiring has already taken place. So the answer will be option A. Coming to next question. Some species of plants and animals invade new habitat across continents. And an interesting problem is to know how or why these invaders are successful. One hypothesis is that invaders carry parasites that do not harm them but infect native species in the new habitat. So we all know that plants and species invade new habitat across continent, and interesting problem is that these invaders, they become successful in the new continent. So they have told about some hypothesis, that is that these invaders, they carry parasites which do not harm them but they infect in the native species in the new habitat. So what did Wilson, Sachs and colleagues do? They tested using this hypothesis using invasive beetle Harmonia that carries microsporidian parasites, and it invades population of native beetle Coccinella. So Harmonia is the invasive beetle and it is having the parasite Microsporidium MSP, and this parasite, it is not harmful to Harmonia but it kills the beetle, invades the population of beetle Coccinella. So below you see a graph from their experiments showing the fraction of Coccinella beetle surviving as a function of time. HAMSP is live microsporidia from Harmonia hemolymph. CS is coccinella, PBS is phosphate buffered saline, N is sample size. What can you infer from these data? So these are the number of individuals and days after injection. So this straight line graph, it is for Harmonia microsporidia from Harmonia hemolymph in coccinella. PBS, this is this graph, this dotted line, it represents the PBS control in coccinella, and this is for heated HAMSP in coccinella. Now, microsporidia cause substantial mortality in native coccinella. That is correct, because from 100% it has decreased drastically to around less than 10% over here. Microsporidia are susceptible to heat. That is why when once the number of individuals, they decrease from 100% to 70 or 80% only, showing microsporidia are susceptible to heat. If it was resistant to heat, then you would have got the graph as given in this graph. You would have got a graph similar to this, but that is not the case. So both A and B are correct. Answer is option D. Then next one. In a recently published study, Tinalion and colleagues allowed 114 independent replicate populations, that is lines, of E. coli to evolve in the laboratory at high temperatures for 2000 generations, and then sequence the entire genome of one bacterium from each evolved line. The figure shows a subset of their data for each of the three types of mutations. The fraction of mutations that were common to 1, 2, 3, 5, 6 to 10 or more than 10 of the independently evolved line is plotted. Based on the graphs shown, which of the following is not true? So E. coli, they grow for 2000 generation at 42 degrees Celsius temperature. Then genomic DNA was isolated, then analyzed for mutation by comparing with the control. Control is the one which is grown at 37 degrees Celsius. And they analyzed for these mutation, that is IS insertions, duplications and large deletions. So option A, approximately 60% of gene duplications are unique to single evolved line. If you see this one, in duplication, this graph, number of affected lines is approximately 60%. So option A is correct. They have asked for the one which is not true. Mutations unique to a single evolved line are more likely to be duplications. Mutations, yes, there can be duplication, but IS insertions are also showing approximately 60% value. So this statement is not correct. B is not correct. Most large deletions are shared between many different lines. This is for the large deletion. They are shared between many different lines. This one, greater than 10. That is correct. Then the probability of observing a shared mutation between 2 to 5 lines is very low. If you see the second graph, this graph, IS insertions graph, the bar from 2 to 5 is short. Similarly, for duplications graph also 2 to 5 is short. For large deletions also 2 to 5 is short. So the answer here is mutations unique to single evolved lines are more likely to be duplications.